And then we have the membrane proteins that we chatted about before. We have integral membrane proteins that can act as transporters or enzymes or receptors. Like I said before, these proteins can act as channels to transport ions through the membrane. They can be receptors that are activated by a ligand to promote chemical reactions inside the cell. And these proteins are permanently embedded in the membrane. Peripheral membranes membrane proteins, on the other hand, are mobile. They can function to link cells together, change cell shape, or act as an enzyme to go on to promote chemical reactions in the cell. A ligand could attach to the outside of the cell onto an integral membrane protein, change the shape of that membrane protein, which would release the peripheral membrane protein that's attached, and then that peripheral membrane protein could go on to do a function downstream, like cause a different chemical reaction. So one classic example is that of neurons. They have receptors on the outside of their cell that respond to chemicals outside the cell and release their own chemicals to go on to elicit a different reaction elsewhere. All right, so now we can talk about the different types of channel proteins. There are 3 different types that we'll chat about. Non-gated ion channels are always just going to be remaining open. So they allow certain molecules to pass through based on concentration gradients. One example is a potassium channel that allows potassium to leave the cell down its concentration gradient. So there's more potassium in the cell than outside, so it just freely goes through the cell, outside the cell. A voltage-gated ion channel, on the other hand, is one that opens or closes based on an electrical charge change of the membrane. For instance, if the resting membrane potential changes in this cell membrane, the sodium channel will open and allow it to pass through the channel into the cell, changing the charge of the cell, right? Because you're having a positive ion enter the cell, making the inside of the cell more positive. Potassium, so there's a classic example of this when these 2 channels are working together, a non-gated ion channel and a voltage-gated ion channel in the case of neurons. So potassium will leave the cell. We have a positive ion leaving the cell, creating a negative charge on the inside of the cell, and then that makes the membrane change charge, changing the conformation or the structure of this protein, which thus changes the function by opening it, allowing a sodium ion to selectively go through and make the or return the inside of the membrane to a more positive charge again. So we'll get into this a little bit more into detail when we talk about the nervous system, but for now, just kind of understand the difference between these 2 types of receptors. And then we also have a 3rd type of receptor. This type of receptor is called a ligand-gated channel. We've already discussed this multiple times throughout this presentation. This type of protein is a receptor for a ligand, or a substance that, once bound to the receptor, elicits a change in the receptor. So it changed the structure of the protein once it bound, and then it changed the function of the protein by opening this channel and allowing sodium to go through and change the charge on the inside. So again, a ligand is anything that can bind to a receptor and change its structure, thus changing its function while bound. Alright, so now we can talk about the glycocalyx that I mentioned earlier. This consists of glycoproteins and glycolipids. So the glycoprotein is a carbohydrate attached to the protein, like an integral membrane protein, and the glycolipid would be attached to the phospholipid head. Okay. The glycocalyx gives the membrane a sticky, sugary coated surface, which allows allows the cell to recognize other cells, and each person's body has like a unique glycocalyx. So these are products of the person's genetic makeup. They give that individual cells an identity and are the primary way that an individual's immune defense cell knows when to attack a foreign pathogen and also not its own cells. So it aids in recognition of other cells. It's also the reason that organs that are donated by another person could be rejected, as the glycocalyx lining the cells of the new organ are not the same as the recipient's own organ cells. So now we can discuss substances that move through the phospholipid bilayer. As we say, or as I said before, the selective permeability of the phospholipid bilayer allows certain substances to pass through freely and others not so much. So lipid-soluble molecules can pass through the lipid bilayer because They're usually nonpolar and small, whereas water-soluble molecules need help to get through. So we'll discuss different transport mechanisms in more detail, but for the purposes of this slide, you can think of passive transport not requiring energy and active transport requiring energy. So diffusion is basically high concentration gradient to a low concentration gradient. So something that has a lot of it on the outside, such as glucose, this is simple diffusion, such as the potassium that we were talking about before. That would be moving through a protein channel, but we can talk about simple diffusion like oxygen or carbon dioxide or alcohol being able to move through the membrane freely because they're lipid soluble. Whereas facilitated diffusion, they need a protein. So this is kind of like the potassium I was just going to say for simple. It's not that. You would need a protein for the potassium because it's charged. So it would be able to freely move down its concentration gradient from a region of high concentration to low concentration. Or potentially even need that protein to open based on that charge or something, but that would still not require energy. Active transport, on the other hand, is usually moving from a region of low concentration to high concentration. It can be from high concentration to low concentration, but the example that we'll talk about will be moving things against their concentration gradient and using energy to do so. So just starting off with simple diffusion, since the membrane is lipid-based, small substances that are lipid-soluble can pass through the cell membrane down their concentration gradient. And I keep saying that word concentration gradient, or phrase. What does that even mean? Well, a concentration gradient is basically saying that there is a region of higher concentration of the substance than another region. It's the difference in concentration of a substance across a space. So molecules will spread or diffuse from where they are more concentrated to where they are less concentrated until they are equally distributed in that space. This is moving down the concentration gradient. So if you see this over time, there is more small uncharged molecules, non-polar, like oxygen or carbon dioxide or alcohol, on this side of the membrane than this. And since they can pass through, over time, they will equalize. They will diffuse through freely until they are equal on both sides. If it ends up being more on this side for whatever reason, then it will diffuse through back this way. But it's simple diffusion from a region of high concentration to low concentration. So in the case of this cell membrane, we have those small non-polar lipid-soluble molecules like oxygen or carbon dioxide that can move through the membrane freely. They do not need a protein. Oxygen will typically move into the cell because oxygen is used in metabolism. Carbon dioxide will usually move out of the cell because it is a byproduct of metabolism. Inside the cells, we gotta get rid of it. And again, simple diffusion does not require energy. It is passive. Next, we can discuss osmosis. So water can also move freely across the membrane of cells, either through protein channels or by slipping through the lipid tails of the membrane. Osmosis, again, is the diffusion of water through a semi-permeable membrane. If you look at the picture here, the red line has holes in it, which allows water to move through it since it is small and uncharged. And, well, I shouldn't say it's uncharged. You can just say water can move through it since it's small and the solute is large. Okay, so this line is selectively permeable. It is allowing the water through but not the solute. Osmosis occurs when there is an imbalance of solute concentration in a cell versus out of the cell. Tonicity refers to a relative solute concentration in a given solution. A solution that is said to have a higher concentration of solutes than another solution is said to be hypertonic. The cells that are in a hypertonic solution will shrink or shrivel as water leaves the cell via osmosis. So water molecules tend to diffuse into a hypertonic solution. So the water is leaving the cells into the hypertonic solution. There's more solute outside the cells, and so the water is going to go to that region of higher solute concentration. So then in the other side of things, we can talk about hypotonic, which is a lower concentration of solute than another solution, and water will tend to diffuse out of a hypotonic solution. So a cell in a hypotonic solution, So there's more solute inside the cell than outside. There's less solute inside or outside the cell than inside. Water will diffuse into there to try to equalize that. And eventually the cell will burst because there will be too much water and it will swell. So in this picture over here, the solution on the left side has a lower concentration of solute than the right side. So the water is going to move from a region of higher solute concentration, or it's going to move into the region of higher solute concentration. So there's lower solute concentration over here. It's going to go from low to high solute concentration. Okay? Thinking about it in terms of the solute, water moves from low concentration of solute to high concentration of solute. If the water is moving from a region of high concentration of water to low concentration of water. Okay, so low concentration of solute to high concentration of solute, high concentration of water to low concentration of water. Whichever way makes the most sense for you to remember, that's hypertonic versus hypotonic. Okay. So again, this process of osmosis is passive, meaning that there is no ATP consumption used. No energy is used. A critical aspect of homeostasis in the body is trying to maintain an internal environment that is isotonic, meaning that there are equal concentrations of solutes in and out of the cell. When this balance is disrupted, various organ systems are going to attempt to compensate for this, such as the kidneys. Okay, now we can talk about facilitated diffusion. So, large polar or ionic molecules, which are hydrophilic, cannot easily cross the phospholipid bilayer. Very small polar molecules, such as water, can cross because it's small enough and they can simply diffuse due to that small size. We saw that in osmosis on the previous slide. But charged atoms like sodium or potassium, or molecules of larger size like glucose, cannot cross the cell membrane by simple diffusion, since the charges are repelled by the hydrophobic tails in the interior of the phospholipid bilayer. So solutes that are dissolved in water on either side of the cell will tend to diffuse down their concentration gradients. But because most substances can't pass freely, they need a protein to help them do that. So that's kind of shown here. You can see that this substance can't get through. It needs the help of a protein in order to get through. And this is called facilitated diffusion, which is the diffusion of substances through the lipid bilayer with the aid of a protein. And the reason they can't get through is due to their size, or their charge, or their polarity, and that's kind of shown here. Although glucose can be more concentrated outside the cell, there's definitely more green blobs out here. It can't get through because it's not nonpolar. It is hydrophilic, okay? And so it can't get through, and so it needs the help of a protein to get through. There are other solutes that have to undergo facilitated diffusion too, not just glucose, such as like amino acids or ions. So we were talking about the potassium pump earlier, or channel, I should say, potassium channel. Also waste products. So because facilitated diffusion, again, is a passive process, we don't have to use energy at all.